At this point, we focus on certain select topics which are pertinent and important for various aspects covered in the course so far. In the process of tissue healing following injury, the stage of proliferation repair is of paramount importance because it is during that stage new tissue is formed. Formation of new and functional tissue around our implant materials and devices is the desirable, successful outcome to which we, biomaterials, biomedical engineers, aspire. The outcome of new tissue formation depends exclusively on the presence and function of cells, the living components of tissues. I will start with a reminder of the definition of a biological physiological tissue which we saw earlier in the course. A physiological biological tissue is an organization of various cells and their respective extracellular matrix. The extracellular matrix has components such as proteins, proteoglycans, etc., and it is an environment which, in addition to biochemical conditions, provides biophysical stimuli which the cells need in order to survive and to function. As a result of this combination, we have unique characteristic structures in each tissue of the human body, and in addition, each tissue has specific biological, physiological functions. As far as the cells are concerned, some pertinent facts are the following. The average human body contains about 100 trillion cells. In addition, the human body has more than 200 types of differentiated cells. Those are the cells which have reached the ultimate stage of their differentiation process. Therefore, they are not stem cells anymore. As listed on this slide, cells have many functions. Among them, the one which is of paramount importance regarding the proliferative repair stage of the wound healing process is, of course, cell proliferation. Despite the large number of cells, as well as the many different types of cells in the human body, a major concern and limitation is their proliferative capacity. In this respect, not all cells in the human body are created equal. For the reasons stated in the introduction, Lecture 19 addresses the proliferative capacity of human cells. In addition, this lecture includes a short introduction to a recent development which has provided great potential and possibility of solving the problems of the limitations of the proliferative capacity of human cells. And that, of course, is the use of stem cells. In terms of their regenerative capacity, human cells are classified in the following categories. Labile are the cells which proliferate throughout life without humans being aware that these processes are taking place. Specific examples include epithelial cells, lymphoid cells, hematopoietic cells in the bone marrow, the gastrointestinal cells. Another category are the so-called stable or expanding cells. these cells retain proliferative capacity, but they don't do so, proliferation-related functions, without an appropriate stimulus. They need to have either a biochemical or biophysical stimulus in order to proliferate. Examples of such cells include most of the cells in the human body, cells in the liver, kidney, of course the ubiquitous fibroblasts, smooth muscle cells, osteoblasts, chondrocytes, vascular endothelial cells, etc. The third category is the so-called permanent or static cells. These are the cells that cannot reproduce themselves after birth. And examples of those cells are the ones which are part of the nerves, the skeletal muscle, the cardiac muscle. These are cells of very important organs in the human body. And it is well known that when these organs are injured, their recovery and their healing is problematic and usually is not completed. The lack of proliferative capacity or limited proliferative capacity from cells which are the major components of important tissues and organs of the body, such as the heart, the nervous system, the skeletal muscle, has resulted in many clinical problems till very recently, when stem cells became available, and because of their important potential of replacing any other type of cell in the body, have given hope that we, or especially our colleagues in the medical care field, can overcome the limitations of the proliferative capacity of cells in the important organs and help the patients who suffer from such injuries. This slide gives you a schematic illustration of the regenerative potential of cells from various human tissues. In addition, some different words are used to describe what I gave you as the three groups classifications of cells in the human body pertinent to their regenerative potential. Notice that in the middle we have a green wedge. Above the green wedge are three arrows which indicate regions of the regenerative potential, which is now described in terms of cellular turnover and regenerative potential. On the left-hand side, there is a region of cells which have high cellular turnover and high regenerative potential. If you look below the green wedge, you see a group of cells, and under this category you see the blood cells, the hematopoietic cells, epithelium from various tissues. In the middle part, going now above the wedge, we have a group of cells which have low cellular turnover but high regenerative potential, depending on what the stimulus to which these cells are exposed is and what is the bioactivity of that particular stimulus. Underneath this particular group, you have the vascular endothelium and a few other cells. The extreme right group, designated as low cellular turnover and low regenerative potential, has underneath the wedge a group of cells which, as I mentioned, have an impossible task in terms of their proliferation. They do not proliferate once the tissue or the cells are damaged, and those cells include the ones from the brain, therefore the central nervous system, the heart, the spinal cord. The interesting and important thing in this illustration is this green wedge, which now brings about into our discussion and into the picture and turns around this situation of the impossibility of healing injured tissues whose cells do not have regenerative potential. This green wedge, of course, is that contribution of the stem cells to repairing tissues and maintaining homeostasis in the human body. These aspects will be discussed in some detail in the slides that follow. I will give you a short introduction to stem cells because I know that all of you who have taken one of the prerequisites for the biocompatibility of materials course, specifically cellular engineering, have covered this particular topic to a greater extent in that course. Let's start with the definition of stem cells. These are the cells that have not yet differentiated terminally, and because of that, they have the potential to give rise to various other types of cells. Among the characteristic properties of stem cells, which are important for the purposes of our course and distinguish them from other cells in the human body, are the following. The stem cells have the ability to self-renew through cell division, and they can do that for long periods of time. A stem cell, therefore, has the ability to make identical copies of itself, as shown in this particular cartoon. The green circles indicate stem cells. We start with one, and of course, the one can split and become two cells which are identical copies of the mother cell. Another characteristic property of the stem cells is that they are non-specific cells. They are pluripotent. That word pluripotent indicates that they have the potential to become cells different, many different cells. The potency of the stem cells is summarized on this particular table. The column under the title potency gives some of the terminology which is used to indicate the potency of the stem cells. The middle column gives you the differentiation potential, and the right-hand column gives some stem cell examples. Starting with totipotent, these are the cells which, from the point of view of differentiation, can differentiate into cells for an entire organism, and then the pluripotent cells are the ones which are present in embryonic stem cells. Multipotent stem cells have the capacity to give rise to many different types of cells. An example is the mesenchymal stem cells, which are found in the bone marrow. Oligopotent means that these cells have the capacity to become only a few types of cells, two or more. Example of such stem cells are the neural stem cells. Last but not least, unipotent are single linear cells, and an example of that type of cell is the spermatogonial stem cells. The third important characteristic of stem cells is that they can differentiate. They can become cells which are pertinent to other tissues and organs of the human body, and that particular differentiation or change is accomplished by their exposure to either specific physiological conditions or laboratory conditions. Such conditions can be biochemical or biophysical. This ability to form other types of various tissues is very important and unique characteristic of the stem cells. The important thing to keep in mind is the following, that as this process takes place, the mother cell differentiates into one type of cell which is similar to its own. Therefore, one of the daughter cells remains a stem cell, keeps that phenotype, while the other one, as indicated in this cartoon with the orange hexagon, is the type of the cell into which the stem cell has differentiated. Therefore, the orange hexagon represents a differentiated cell which is specific to a tissue or an organ in the human body. An important aspect which proved to be crucial in the survival and function of stem cells is the so-called niche. This is a microenvironment which provides the conditions which the stem cells need to be protected and to survive either in a quiescent state till the appropriate stimulus is received, and then to proceed into the functions which are pertinent to, let's say, their differentiation. An example of such a niche is the bone marrow. This is the place where hematopoietic stem cells and mesenchymal stem cells are present. The hematopoietic stem cells are those which give rise to all the cells of blood. The mesenchymal stem cells are important stem cells which can differentiate into cells which are important in bone, specifically osteoblasts, cells which are important in cartilage, specifically chondrocytes, and cells which are found in the fat tissues of the body, and they're called adipocytes. In the environment of their niche, the stem cells can survive for indefinite periods of time. The balance between stem cells, the other cells which are found in that microenvironment, the extracellular matrix, the chemical compounds such as growth factors, etc., all of these conditions that are found in the stem cell niche are very important because this is assurance to maintain the stem cell phenotype for long periods of time. A warning or caution note is given here, namely that when the stem cells are isolated, purified, and cultured, these are traditional techniques now in cell culture laboratories, the processes involved separate the stem cells from their niche. As a result of that, the cells may be affected and not function as they do in the body under normal physiological conditions. For these reasons, the task that we who are cellular bioengineers have is to provide the stem cells as well as the differentiated cells later on with conditions which simulate, imitate, duplicate their normal niche environment, and thus assure that the stem cells will survive and function as they do in the body. They will survive and function in the laboratory and under the conditions we need them for biomedical applications. The process of differentiation of a stem cell to provide a differentiated cell which is specific to a given tissue takes place in several stages. starting with progenitor or precursor cells, which are found in a specific niche in the body. One of them, and the first one, which was identified as the source of stem cells, was of course bone marrow. Then committed stem cells to a particular phenotype. By that we mean a particular type of differentiated cells from a specific tissue, terminal differentiated cells, and last but not least, the mature differentiated cell. This particular process schematically is shown in this cartoon, starting with the green circles of the stem cell, which now differentiates, always provides a stem cell daughter, and then the various stages of the differentiation, which are shown here as orange hexagons, and you can see the various stages till the end. And of course, at the end, we have the burgundy pentagons, which illustrate the differentiated cells. Depending on the origin, stem cells can become blood cells, cardiomyocytes, chondrocytes, endothelial cells, hepatocytes, neurons, osteoblasts. The important thing to notice on this limited list of cells from the human body is that some of these cells can be present and or can be provided through normal physiological procedures. But as we said, certain tissues and cells in those tissues, like in the heart, therefore cardiomyocytes, or in the nervous system, neurons, they don't have that capacity to proliferate, and therefore the organs of which they're the important dominant component, they cannot heal themselves following injury to that tissue and organ. The history of stem cells is relatively recent. Mouse embryonic stem cells were isolated for the first time in 1981. Human embryonic stem cells were isolated in 1998. The important facts regarding embryonic stem cells are, first of all, that they do not exist in the body after birth. They are isolated during the early stages of the embryo formation and life, and they can be expanded in vitro in the laboratories. They're pluripotent stem cells, therefore they can differentiate into many other types of cells. And because of that particular promise, they have great potential for biomedical engineering applications, specifically in tissue engineering, regenerative medicine, and regenerative engineering applications. The mouse embryonic stem cells proved to be a very important model, which allowed the researchers working in this field to perfect techniques of cell isolation, study the early stages of embryonic development, and also study and understand the various stages of cell growth and differentiation. An additional experience and knowledge which was provided from this model was when these cells were obtained from genetically modified mice. Therefore, important information was provided regarding what is the big difference between health and various disease states. Unfortunately, the major limitation regarding applications of embryonic stem cells is the fact that these cells cannot be used for clinical applications. Of course, it's out of the question that they can be used in transplantation, but even the lessons that were learned from studying embryonic stem cells, because of the difference in the species, those lessons cannot be directly translated into improving enhancing the knowledge that we have about the human body. The limitations of embryonic stem cells from various species other than humans provided the rationale for research related to human embryonic stem cells. These type of cells were derived from embryos which were created but were not used for reproductive purposes using in vitro fertilization methods. However, concerns regarding human embryonic stem cells arose, and the controversy was regarding the use of human embryos, issues of ethics, first of all, obtaining and then using the embryonic stem cells have been the subject of national debates in this country, the United States of America. Some countries forbid human embryonic stem cells research, but some others do not. Laws and regulations regarding human embryonic stem cells are now covering production of human embryos for research, how the human embryos available from various unrelated medical treatments are used, how to harvest and thus make available the human embryonic stem cell lines, how the human embryos are used in research, the use of whatever lines have already exist and have been established from the past, and all kinds of other concerns regarding of transfer of various parts of the embryonic stem cells for therapeutic cloning and reproductive cloning. The federal laws of the United States prohibiting procurement and use of human embryonic stem cells provided a good reason for researchers to seek alternative sources of stem cells. This research resulted in the discovery of human adult stem cells. The first human adult stem cells were discovered in the bone marrow. These cells are not very common. They are found in very small numbers, specifically 1 in 100,000 cells in the bone marrow. And in addition, as humans get older, the number of the stem cells is reduced. In addition to the bone marrow, stem cells were discovered in many other organs and tissues. For example, at the bottom of each piece of our hair, there is a follicle, and there researchers discovered one stem cell per piece of our hair. Another source of stem cells turn out to be the blood in the umbilical cord, which is a tissue discarded after the babies are born. The adult stem cells are undifferentiated cells. Of course, they are found, as I mentioned, in relatively small numbers among differentiated and specialized cells of various tissues and organs in the human body. Adult human stem cells can differentiate, and they can form all kinds of different cells, differentiated cells from tissues and organs of the human body. The differentiation of the adult human stem cells is restricted sometimes into specific lineages or types of cells from specific tissues. For example, the hematopoietic stem cells, which are found in the bone marrow, can differentiate into all types of blood cells only. The mesenchymal stem cells, which is another type of stem cells in the bone marrow, can differentiate into bone, cartilage, and fat cells. The discovery of human adult stem cells was the impetus for a totally new field of biomedical research. For this purpose, the stem cells had to be isolated from the pertinent human tissues. Of course, that particular isolation started with tissues which were easily accessible. For example, the bone marrow. Hematopoietic stem cells are present in the bone marrow, but in addition, they are present in the placenta and the blood in the umbilical cord. Both are tissues which are discarded after the babies are born. In addition, mesenchymal stem cells are present in the bone marrow of the iliac crest and on the head, the top of the femoral bones. Another tissue which proved to be a very good source for mesenchymal human adult stem cells turned out to be adipose or fat tissue. At the beginning of the field, researchers needed to do all this work, namely to obtain the pertinent tissues, develop techniques to extract the stem cells, develop techniques to proliferate the stem cells, make sure they have techniques that do not affect the functionality of the stem cells, and last but not least, use them for their research purposes. Nowadays, all these types of human adult stem cells can be obtained commercially. There are several companies which do only that, namely isolate, proliferate stem cells, adult human stem cells, and they make them available to researchers, of course, for an appropriate fee. As any researcher who works in cellular engineering finds early in his or her training, a very important aspect regarding the isolated cells is to characterize them. For example, even if one starts with bone and isolates cells, one cannot just claim that the isolated cells are osteoblasts. Keep in mind that every tissue has more than one type of cells. Some of the types of cells are predominant, but there are other cells which, in the terms of the isolation procedure, we may call them as contaminants. In a similar fashion, the isolated adult stem cells need to be characterized. And in this case, there are certain membrane markers which need to be identified, and they have been already known because of extensive research in this particular field. What is needed in the case of adult stem cells is to show that the isolated cells have certain specific markers, therefore presence of specific markers, and at the same time absence of other markers. This combination of presence and absence assures the characterization of the isolated stem cells and gives additional information regarding of how pure the particular preparation, the particular isolated preparation is. The markers on the membranes of the stem cells are referred to as clusters of differentiation, and they're abbreviated as CD. In the case, for example, of hematopoietic stem cells, extensive research has shown that in order to prove that a particular isolation contains hematopoietic stem cells, you must show that all these compounds with the plus signs next to each compound are present, positive. In addition, last entry, a CD38 should be minus, negative. That is the important thing. The combination of the positive present and the negative absent. In the case of mesenchymal stem cell, STRO1 has to be positive, but CD34 must be negative. I must add that more markers have been identified recently regarding the mesenchymal stem cells, therefore this particular list is not complete. If you are the researcher who isolates adult stem cells, it is your responsibility to do all these characterization experiments, and let's say if hematopoietic stem cells is what you are hoping to obtain, that you show that all these CDs positives are present and the negative ones are absent. An alternative these days, since we have commercially available stem cells, is the following. The companies which sell to researchers stem cells, adult stem cells, guarantee that they have done their homework and they've done this characterization. So when you get the supply of the stem cells which you need for your research, you get a list of all the results of the characterization tests that the company conducted. And you have the list of all the positive CDs and all the negative CDs, and you get assurance that what you have bought are hematopoietic stem cells. Of course, that expedites, I should say, your process of doing the experiments, but this is what you pay for the companies when you buy the cells from them. Not only the cells, but also the cost of the experiments they conducted to characterize the isolated adult stem cells. Determination of the clusters of differentiation, the CDs which are present or absent on the membranes of the isolated adult stem cells is only the beginning of the characterization of these cells. What is also needed is proof that these particular cells do function as stem cells. Many times, techniques which are used for the isolation of stem cells may result in a good harvest of these cells. However, if those cells are not functional, the particular process is not useful for biomedical research purposes. The functional characterization of the isolated adult stem cells requires cells which show that these cells can differentiate to whatever type is needed for a specific tissue. We call that phenotype differentiation. And to form functional tissue, hopefully in vivo eventually, but first in vitro. For example, if you start with hematopoietic stem cells, what people have done is to take these hematopoietic stem cells and introduce them in the bone marrow of animals which have lost their population of bone marrow stem cells as a result of exposure, let's say, to radiation. And this is, of course, a therapy which is used to cancer-stricken patients, and it is an undesirable side effect. They lose the bone marrow cells. So, in animal experiments, they have taken the hematopoietic stem cells after isolation and put them inside the animals, and would like to determine whether that particular intervention, transplantation, resulted in repopulation of the bone marrow, and that result lasted for a long period of time. In terms of the mesenchymal stem cells, researchers have used in vitro laboratory experiments and given the cells specific chemical stimuli, studied their differentiation into cells from adipose tissues, fat tissue, or to cells from cartilage, specifically chondrocytes, or from bone, therefore osteoblasts, and in addition formation of the mineralized matrix. Pertinent in vivo experiments use immunodeficient animals. These are animals which obviously are having a major deficiency of the immune system, and by bringing transplanted mesenchymal stem cells, the studies explore the outcome which now reverted that particular deficiency and cured the animals from that particular medical problem. So far, lecture 19 was a short introduction to stem cells, historical background, isolation, characterization of these cells, and the great potential and promise in clinical biomedical applications. The rest of the lecture will address some aspects which are pertinent to the use of stem cells from the engineering perspective. For this purpose, stem cell use in clinical and biomedical engineering applications will be considered. The promise and potential of stem cells in clinical and biomedical engineering applications remain high. In the interim, biomedical scientists and engineers working in the field have identified certain needs which must be addressed and improved. Starting with the need to optimize the culture methods. Human adult stem cells are present in very small numbers in source tissues. In order to be used for applications, a larger number of these valuable cells is needed. For this purpose, these original cells must be cultured in vitro and expanded, proliferate. These methods, culture methods, are time-consuming, labor-intensive, and so far they've been used exclusively in research laboratories and therefore provide a relatively small number of cells. The techniques are not suitable for large-scale cell propagation, which might be needed in order to bring this type of cells to clinical use, to the market for clinical use. Another aspect is the need for optimizing the cell culture protocols. Various cells are cultured in the laboratory in the presence of serum from animals, usually cows. Serum, remember, is the liquid part of blood after the blood was clotted, and therefore we had that serum has no fibrinogen and fibrin. The use of serum from animals expedites the process of cell cultures in the laboratory, but even when these expanded cells may be used in humans, it is possible that the result may be rejection because the immune system in the recipient patient is activated. Last but not least, the cell culture techniques do not give us 100% homogeneous cell type populations. Remember, every tissue has a predominant one or two types of cells, but it has many other cells which are part of the normal biological, physiological tissue. When the cells which one is after are extracted and cultured, one can never obtain a homogeneous, 100% homogeneous population. For example, when we extracted osteoblasts from the rat Calvaria, we could get a population of approximately 90 to 95% osteoblastic. The rest were what we call contaminant cells, and they were mostly fibroblasts. This particular difficulty of obtaining 100% homogeneous cell types is an issue that needs to be improved for future applications of adult stem cells in the clinical environment. So far, animal experiments, the next step after in vitro cell using experiments, these animal experiments have used relatively small size animals. These are usually rats and rabbits. Before successful results, which may lead to clinical applications, is the need to transfer this particular knowledge and experimental techniques using large animal models. The current challenges which need to be addressed, especially by biomedical engineers working in the field, is the need to expand the undifferentiated cells. To increase the number of undifferentiated stem cells in vitro is also known as to maintain the stemness. Possible solutions regarding how to achieve this particular outcome is, of course, development of new cell culture conditions, and last but not least, an area where biomedical engineers are making major contribution, the use of bioreactors. Another important aspect that needs to be successfully addressed is the development of approaches to control in vitro cell differentiation. Since stem cells are pluripotent and multipotent cells, it is not easy. As a matter of fact, it's very difficult to obtain a homogeneous one type of cell population when these cells differentiate. One particular example is the following. Stem cells, mesenchymal stem cells, have the capability of becoming either cells from fat tissue, adipocytes, or from cartilage, chondrocytes, or from bone, osteoblasts. In the petri dish, in the laboratory, sometimes these cells take off, and in one and the same population, even under what the researcher thinks they're control conditions, they become a little bit of everything, while the researcher may want only chondrocytes or only osteoblasts as the population, the differentiated population of those cells. Possible solutions to this particular challenge include to change the biochemical stimuli, which of course are provided in cell culture medium, by including select and appropriate growth factors, cytokines, and other proteins. Other possible solutions which are pertinent involve changes in the environment of the cell culture, to use 2 versus 3 dimensional cultures, co-cultures, for example, in the tissues, since we have more than one type of cell, perhaps the cells would like to have in their neighborhood the other types of cells which are present in the physiological tissue. And last but not least, something which only biomedical engineers can do, and they have contributed to this field, is to provide select and appropriate biophysical stimuli. And those stimuli, of course, include mechanical stimuli, shear stress, tensile stresses, pressure, or electrical stimuli, etc. The field of stem cell research is very active and productive. Despite several important contributions, still many pertinent issues remain unanswered and require current and future scientific attention and work. For example, the mechanisms which underline the stem cell differentiation needs to be understood much better. For that purpose, we need additional research in this particular field. We need to understand how to control stem cell differentiation in order to be able to apply this important group of cells for successful clinical applications. Stem cells currently are of central importance in various cutting-edge fields in biomedical applications, specifically tissue engineering, regenerative medicine, and new therapeutics. All of these fields are very active as we speak. Last but not least, the methodologies which are developed for stem cell related research eventually need to be translated from the lab into the clinics. A particular phrase which is used for this kind of translation is from the research bench to the patient bed. This particular aspect is of crucial importance for clinical applications. Scientific advances and contributions are important, but for the biomedical needs, we need to eventually have all these results translated into what our colleagues in the healthcare field can use in order to help patients, namely humans and animals. In closing lecture 19, I would like to acknowledge the assistance of former UTSA graduate student Dr. Courtney Creasy, who assisted me with some of the slides which I used in this presentation. The information I used in this lecture came from the following chapter with the title Stem Cells, chapter number 1, in a book edited by colleagues in the Netherlands. The book's title is Tissue Engineering. This is the end of lecture 19 with the title The Proliferative Capacity of Human Cells, Stem Cells.